Okay, so this morning then I'm going to talk about the latest features in CCP4 6.3.0. We've some exciting updates to the software and some new things available, and I'll try and cover as much as I can in the half an hour. Just before I start to talk about the software, I give a brief introduction to who CCP4 are. So CCP4 stands for Collaborative Computational Project number 4, and it's one of several CCPs that was set up in the UK to advance and support scientific software development in various fields. CCP4 itself was set up in the late 70s. It was started in 1979 specifically, and it was meant to bring together the leading developers of software in the field of protein crystallography throughout the UK. And the main aim was to assemble a comprehensive suite of software to satisfy the requirements of the various crystallography groups throughout UK and later on throughout the world. Just to show you where we are, the CCP4 main group is hosted by the Science and Technology Facilities Council, which is down near Oxford in the south of England. We're in what's called the Research Complex at Harwell, which is next door to the National Synchrotron for the UK, Diamond. And the Research Complex basically hosts any groups that have a vested interest in what takes place inside the synchrotron. We're funded by two of the research councils in the UK, the BBSRC and the MRC, the Medical Research Council. And we also receive some industrial funding through the sale of commercial licensing. The core group itself, which is based here in Oxford, what we do is we maintain and support the software suite. We do some application development and infrastructural software development. We do a lot of collaboration with the Diamond Synchrotron people on the deployment of software on their MX beamlines. We're also involved heavily in educational outreach through workshops and conferences and now webinars. And we maintain such things as the CCP4 bulletin board and the CCP4 help desk. Outside of Oxford, we have a large group that are based in Cambridge. One set at the Laboratory of Medical Biology, where programs, data processing software such as MOSFilm and Aimless are developed. Also, Garb Murshidov is based there, the developer of RefMac and various other software such as BALBES. And very recently, Paul Emsley, the developer of Coot, has moved to the same institute. As well in Oxford, in the university itself, we have the Phaser group, who contribute quite a lot of software to CCP4 as well. Further north in York, we have another major group, a series of software developers working on such things as CCP4 MG, the molecular graphics software, Buccaneer, and things, density modification programs such as Parrot. And as well as that, there's a new project to develop a new GUI for CCP4, and that's being developed at York as well. Beyond that, there are several other groups scattered around. One notable group is the group that develops the Crank software in Leiden in the Netherlands. So, we like to keep track of where CCP4 is used, and this is a nice plot of the world showing where our licenses are. And as you can see, the major markets are in the US, Europe, and Japan. But more recently, we've seen more licenses in places like China, India, and South America. The red dots here indicate where we run schools or workshops, and we use this license map as a guide as to where we should go for these training events. I'll talk more about those a bit later on. So onto the software itself. The CCP4 suite is a comprehensive suite of software for doing protein crystallography. We have new versions of the suite released about once every 18 months. These usually include new programs, any major updates to existing programs, and other features such as changes to the CCP4I interface itself. And just very recently we've had a new feature made available where revisions to current releases can now be installed via what package we've developed called the CCP4 Updates Manager, which allows any user to get access instantly to the latest software. And I'll show you a bit more about that a little later on. So if we look at the standard pipeline from crystallization through to deposition, CCP4's software basically fits into the area from processing the initial X-ray diffraction images right through to structural analysis. And we have quite a comprehensive set of software tools for all of the stages in between. In addition to that, we've quite recently made a new collaboration with the people in Hamburg who developed the ARP-WARP model building software. You can now get ARP-WARP installed as part of the CCP4 installation. And then in those other areas outside of our core area, we have associated projects such as PIMS, which I understand is going to be distributed through SBGrid in the near future, if not already. PIMS is a laboratory information management system. There's also a new next generation data collection and data processing software package being developed called DIALS as part of the BioStructX pan-European project. And then at the end, for structure validation and deposition, we have strong ties with the PDB Redo group from the Netherlands, some of whose software will be made available through CCP4 in the near future. So the current release of CCP4 is version 6.3.0, which came out roughly about 3 or 4 months ago. It included a lot of new programs. And as I mentioned, in collaboration with the Hamburg people, you can now get ARP-WARP rolled with the CCP4 suite. It also had updates to several major programs such as Phaser, RefMac, and MOSFilm. And there's some new infrastructure developments such as the Downloads Manager and the Updates Manager. So I'll go through some of the existing software and some of the new stuff, and I'll step through the interface menu just to highlight where you can find each of these packages. So the first point is data reduction and analysis, and in that area we have iMOSFilm and MOSFilm, iMOSFilm being the new graphical front end to MOSFilm, and we have a new version of that with I think the main addition has been better support for Pilatus images, and also they've done a lot of bug fixes as well. And the other major software here is Pointless, which is for the determination of point group and possibly space group. And there's also a new package called Aimless, which is a replacement for the old Scala scaling software. It's faster, it performs better, and generally it's all around better. So I recommend that you use Aimless now rather than Scala, although Scala is still distributed with the suite. Other than that, we've got the latest version of ZIA 2, the automated data reduction software, which is designed to take you from X-ray, automatically process X-ray images and go right through to generating emerge reflection files suitable for phasing. We use it sometimes at our workshops, and in many cases we find that for reasonable data it will do a better job than what the students are capable of doing by hand.
Ronan, for ZIA, do you use XDS as an integration engine, or what's being used? Because ZIA is just a shell, basically, right, for processing.
Yeah, it wraps around XDS and MOSFilm, so you can use one or other of those. XDS can work slightly better in some cases, I believe. But if you have either of those, you can use it. A new pipeline that we've just introduced is a collaboration with the Diamond people. It's called Dimple. It's a basic, simple difference map pipeline designed to take you from the initial images through to giving you a difference map in as quick as possible a time, basically for the purpose of seeing whether or not a ligand has bound to your protein, and that's now included in the suite. Oh, hang on. Okay, so moving on to phasing then. For experimental phasing, we have the automated pipeline Crank. It automates the process to go from heavy atom location right through to model building. And we also have Phaser EP, the automated experimental phasing pipeline from the Phaser people. It will currently do SAD phasing, and you can also use it to combine MR with SAD phasing. For molecular replacement, we have the traditional version of Phaser for molecular replacement, the maximum likelihood method approach. We have currently we're up to version 2.5.2. And we also have MolRep, the molecular replacement pipeline from Garib Murshidov's group originally. And it has some nice additional features, such as being able to do a phase rotation translation function, and also an option to do a multi-copy search. So what it will do is it will first look for, if you've got multiple copies, it will look for a second copy and then combine those and use those then, like if you've got two, it will use those as a search model for further copies. On the automation side of molecular replacement, we have two pipelines, the first being Mr. Bump, which automates the process from going from an initial search for models through to preparing those models for use in molecular replacement, and then putting them through both Phaser and MolRep. And the latest version of it now has the option to do model building. So you can use ARPWARP or Buccaneer or ShellXE if you have it installed to try and build the structure after molecular replacement and refinement. And this is usually a good way of giving you a clear indication of whether or not molecular replacement has worked. The other pipeline that we have in the suite is BALBES. It's similar to Mr. Bump, except instead it uses it to search for models. It has its own customized version of the PDB database, which includes search models in monomer, domain, and multimeric form. And its emphasis, rather than the sort of broad approach that Mr. Bump takes, BALBES takes an approach where it tries to identify the best search model for use in molecular replacement.
Which one people typically start with, and in terms of the success rate and convergence, do you have any sense? Between Mr. Bump and BALBES for
which
molecular
one?
Replacement? Yeah, which one higher success rates?
Well, I think in terms of success rate, they perform quite similarly, but BALBES has the advantage that of what I'm going to show on my next slide is it has the web service available, so it makes it a lot more accessible to users. The BALBES in the suite is reliant on its database, so that database needs to be constantly updated as new structures are added to the PDB. So for using BALBES, I'd recommend the web service, as it keeps an up-to-date database. And that's what this slide is about. So you can get access to the web service if you just Google basically for BALBES. And you just provide it with your NTZ file and your sequence file, and it will go off and give you your answers back, usually in less than a day. But in terms of comparison, I think, well, the best advice I can give you is to try both of them, really, if you have a difficult molecular replacement problem. So in molecular replacement, search model preparation is always an important thing. It can make the difference between success and failure. So we have several new tools, mostly from the phaser people, for preparing search models. The first is Sculptor, which is a sophisticated program for preparing a model. It can do things like use weighted sequence similarity between the target and your model to decide how best to prune the search model. It will also take into account such things as whether or not in your model residues lie on the surface. So it looks at accessible surface area, and it will downweight these side chains if they are on the surface, as they're likely to be there's more movement there. The other new package from Phaser is Ensembler. What Ensembler does is it will take a set of homologs that you want to use in molecular replacement and then superposition them together and prune them and give you an ensemble model, which in some cases can give you a solution where any of the individual models that go into the ensemble don't. So Phaser itself is quite clever with ensembles. It will weigh up areas that are low areas in the models that have low variance and weigh down those that have high variance. So it can give you a solution in some cases where otherwise you may not. As well as these, we have the MolRep and Chainsaw. It can also be used for preparing models. MolRep, you give it your model and it will do a sequence alignment internally and then prune your model based on that alignment. And Chainsaw, you can give the alignment too. So you can generate an alignment from somewhere else and give it the alignment, and it will prune the side chains of your model based on the alignment. One exciting new development in molecular replacement is a new program called AMPLE, which came about as a result of a pilot project between CCP4 and the University of Liverpool to assess the suitability of ab initio generated search models for use in molecular replacement in cases where there's low homology or there's no obvious homolog to use as a model. So it uses programs like Rosetta to generate what are called ab initio or de novo models, and then these are prepared for molecular replacement. It's found to work well for smaller alpha helical type protein structures, roughly less than 120 residues in length, but it has been known to work for larger ones. I mean, we've had one case where it's worked for a structure up to 250 residues. It requires installation of several third-party applications, such as Rosetta, but I understand if you're a member of SBGrid, then you get most of these applications as part of the SBGrid package.
Ronan, will it work for DNA and RNA molecules as well, or current version is focused on protein structure determination?
At the moment, it's just focused on proteins. There is a plan to extend it further, and that is one of the things we'd like to look at. But at the moment, we just, as part of the pilot project, we looked only at protein structures. It is available as a beta version, so the main idea was to investigate whether it would work or not, which it does, and then as an offshoot, we made available the software we developed through the suite. Just a bit more about AMPL. So what it does is it generates hundreds or possibly thousands of possible decoy models using Rosetta. And then these decoys are clustered to try and get an idea of what the most likely fold is. And the top cluster then is taken on for further processing. And from that cluster, several ensembles are generated. And these are then fed into Mr. Bump, which in turn passes them into Phaser and MolRep for molecular replacement against the target. Moving on then to density improvement, we have two relatively new packages for doing density improvement. Parrot, which uses a maximum likelihood methods approach and builds on the old DM program. It's quite fast and it's fully automated. Alternatively, there's Pirate, which is a statistical-based density modification program. It is quite slow, but in some cases it can work better than Parrot does. And then model building, we have the latest version of Buccaneer, which does chain tracing by identifying connected alpha carbon positions using a likelihood-based approach. It was originally designed for doing model building at low resolution, but it can work also just as well at high resolution. As a natural development from Buccaneer, another program was developed called Nautilus, which is aimed at automatic model building of nucleotide structures into electron density maps. And that's now available as part of 6.3.0. And another offshoot from Buccaneer was Sloop, which is a loop building program. And it looks for gaps in the chain, and then it uses fragments from the Richardson's Top 500 library to try and build the loop into that gap. As I mentioned before, we now have ARP-WARP included in CCP4, or co-distributed. And ARP-WARP is a very comprehensive model building suite. It will build proteins, RNA, DNA. It can do secondary structure finding in density. It can build side chains and loops, and also fit ligands automatically. It also comes with a nice new graphical interface called ARP Navigator, which allows you to see your structure in the map. We have the latest version of Coot 0.7, which came out about a month ago. The main new features there were ORCRANE and Cootaneer can now be used for RNA building. It has the ligand builder has been made to work in conjunction with prodrug for the generation of restraints for refinement. And it also has an interface to JLigand, which I'll say a bit more about in the next slide. One of the things we've been concentrating on here is to try and get wider Linux support for Coot. So one of our guys here has been working on developing nightly builds for Coot in the package management systems of various Linux operating systems. So now you can get Coot more widely on different Linux systems. JLigand then is a new, I think it's been in the suite since 6.2.0, but we've improved it and fixed a few issues. So you can now run JLigand directly from CCP4i. And what JLigand does is it can be used to generate and regularize ligands, and it can also be used to link ligands together and generate link descriptions that can be used then in refinement. Moving on to refinement itself then, RefMac is currently on version 5.7. There's been a slow development recently because Garb has been moving his house from York to Cambridge. But the most recent stuff that's been added is stuff like Cyrus and jelly body refinement. You can do refinement of multiple occupancies. It works better now at low resolution. You can generate restraints from other structures using another program called ProSmart, and then you can feed them into RefMac, and they will help in refinement. It has a map sharpening feature now too. It will do automatic and local NCS restraints. And one of the big new things was a new monomer library, which was much expanded and corrected. It has up to 9,000 structures in it now, and it has better confirmation to the PDB3 format now. There are plans then for new things coming soon with RefMac, either in the current version or in the next version. Improvements to the jelly body refinement protocol, improved use of restraints information, as I mentioned before, with ProSmart. There will be DNA, RNA base pair restraints, sugar pucker restraints. You'll be able to do simultaneous density modification and refinement. It will generate better anomalous difference maps. There's work ongoing looking at the refinement of ensembles. So following on from molecular replacement, where you can use ensembles, the next obvious step is to try and refine against those ensembles and give an averaged set of map coefficients and hopefully give a better map. Other stuff such as multi-imputation and averaging of structure factors to desired resolution. So in more layman's terms, that's a bit like the free lunch option that you get in ShellX. There's sampling of conformational space. Sorry?
So we have a couple of minutes left right now.
A couple of minutes, oh God, all right. Okay, so I'll move on. I'm nearly there anyway. So as I mentioned, there's ProSmart, which is for the generation of restraints for refinement. ProDrug does the same thing. And then another new program, Zanuda, is for the analysis of refinement results and specifically checking that that you have the correct space group. For structure analysis, we have PISA, which is basically for looking at macromolecular interfaces and their interactions. One new thing that's just come out as part of our updates mechanism is the QT interface to PISA. It's very nice. It gives you a very clear view of everything that's going on. There's the new version of CCP4 MG, version 2.6.2. Most of that's just bug fixing and improving reliability. There's a new structure alignment program, Gesamt, which is a replacement for the old Superpose. There's some new utilities, viewHKL, which is basically for looking at reflection data. It combines what you could do with MTZ dump with the old HKL view program. And it's now the default viewer for MTZ files in CCP4I. Just a bit about downloading the suite. It's available as you're on
this one you can skip
SBGrid.
probably because SBGrid people have all of this, the newest version installed right now. So.
Okay, yeah.
But if you guys have any questions about then something isn't working, like with ARPORP, it might not be installed by default, you can, how to contact help at SBGrid, and Ben will take care of that for you.
I'll skip this one as well since this covers the same area. But I mentioned this is the update manager. So if you have an installation outside of SBGrid, then you can use this to update to the latest software. And we do releases of new stuff every 2 to 3 weeks. There's plenty of online resources. There's the CCP4I wiki. There's also the bulletin board. And if you have any problems, you can always email us at helpdesk. One new thing that's just come back after several years is the newsletter. So any of the stuff I've talked about, you can probably find details of it in there, and that's accessible from the CCP4 website. I should mention workshops as well. So we do a lot of outreach.
What was this thing you had on the bottom of the previous slide, CCP4 I2? There was this GUI. We go back one slide.
Yeah, so there's an article in the new newsletter about CCP4 I2. So it just gives you an introduction to what it looks like, what it will feel like. I can show you where to find that if you want at the end of the talk. Workshops. So we do a lot of outreach. They're mainly aimed at helping people with difficult structures and difficult data. There's one coming up in South America in Montevideo in April, and then we have the annual US one in Chicago, and various other ones around the world: Japan, Germany, China. I'll mention specifically the APS one. So that's in collaboration with the GMCA beamline at the APS. It takes place every year in mid-June. People have the opportunity to bring crystals, collect data, and then get help with solving the structures. And then there's lots of software people there. The CCP4, Phoenix, Shellix, ARP, XDS, HKL, and several others are all there and available to help. And watch out for an advertisement for that on the bulletin board in the near future. Finally, then, I'll just mention the study weekend. The study weekend is coming in January. Happens every January in the UK. And as Piotr mentioned at the start, it's about molecular replacement, this coming one. And there's lots of good stuff happens at that. Piotr himself will be talking at it, as I understand. And there's also side sessions where people can come and talk to the software developers, and if they have problems, get help with them. And details of that are all on the CCP4 website. Finally, then, just some acknowledgements. There's a hell of a lot of people contribute to CCP4, and I think I've left an awful lot of people out here. So that's CCP4 6.3.0. Thanks for listening.
Thank you, Ronan, very much. Thank you, and for a very good overview. And if you guys have any questions right now, just send me a text message in the chat window, and I'll moderate to Ronan. A couple things coming here about the automated structure deposition. When you finalize your coordinates, do you guys have anything that will allow you to interface with the protein data bank and make sure that things are properly formatted and complete before you deposit, or what's the best tool here?
We've nothing specifically in the suite at the moment, but we are working with the PDB redo people in the Netherlands, and they have a lot of tools for validation and checking things, and some of that stuff will be going into the suite in the near future. But I mean, that's slightly outside of our area, but you know, we will have that stuff soon.
Okay, and good. In terms of the Coot and CCP4MG, it might be a good idea to do a separate webinar. Maybe we can talk to Paul Emsley or people who are in charge of CCP4MG just to focus on those two applications. In particular, yeah. Somebody was asking if that in past the movie making utility within CCP4MG, it was a bit not fully refined. Do people use it within right now for making movies? Do you have any comment on this in CCP4MG?
Yeah, I mean, I've used it several times myself. It has been a little bit flaky, so to speak. But Stuart, who develops it in New York, is working on improving and making things more stable. But you need to make sure your window is big enough, I think, is one of the problems.